We now understand today that DNA has a helical structure and contains genetic information. Our understanding of DNA has allowed us to make advancements in science in the field of genetic engineering, cloning, and forensic science. We've come a long way since the early beginnings. In this video, you'll learn about the history, the discovery, and the structure of DNA. We'll learn about the transforming principle. It's one of the most classic experiments in biology. This is before anyone had a clue what DNA was, and if it was indeed the thing that stored and carried genetic information. So Griffith, in 1928, experimented with two forms of bacteria cells that cause pneumonia. You have the S cells, and you have the R cells. And if you inject S cells into the mouse dies because it's the deadly form of the bacteria. And if you inject living R cells into the mouse stays healthy. So next, what he did was he would then kill the S cells with heat. He would treat it with heat. And he would then collect the sample and then inject it into the mouse. And the mouse was healthy, so it actually survived the S cells. Okay, so far, the only thing that causes the mouse to die are the living S cells. But now what he's going to do is he's going to mix the heat-killed S cells and the living R cells, combine those two samples, and then inject it into the mouse. So he ends up with this mixture, and then what he observed was the mouse dies. And so obviously there's something strange going on. And then when you perform the autopsy, you would find living S cells in the blood sample of the mouse. So there's something going on here where it's almost like the S cells are coming back to life. And so what later was concluded was the heat-killed S cells were able to transform or deliver its genetic material, and then the R cells became a deadly form of bacteria. So we're on to something with the mouse pneumonia experiment. We know that genetic material is being passed around somehow, but at this point, in the 1950s, scientists still didn't know if DNA or protein carried genetic material. So before I actually get into the details of the next experiment, you have to understand how viruses work, how they replicate, and how they infect bacteria. So these types of viruses are known as bacteriophage. So what actually happens is it lands on a bacteria. That's the virus coming in. And what it does next is it injects its DNA into the bacteria. So it sends the DNA inside. There's the viral genome. And what happens next is that strand of DNA starts to replicate and reproduce. And then now you have all this stuff coming together to create more viruses, more copies of itself. And when the virus is ready to escape, it then causes the bacteria to rupture. It kills the bacteria. And now you have a bunch of these viruses floating around. And they can then start the next process. They can infect different bacteria. And that's how they multiply exponentially. Now it's time to figure out whether it's DNA or protein that carries genetic information. So here's the experiment where you have the bacteriophages, the viruses again, and you're going to grow them in two batches. One batch is grown in radioactive sulfur, that's batch 1 that I'm underlining. And the idea is sulfur is found in proteins. So proteins contain a lot of sulfur. So these viruses were grown in this like solution with a lot of sulfur, and then they come out, and the scientists would just let them go to work by infecting the bacteria. So the viruses would then jump on the bacteria and then infect them. Just like how I showed in the previous clip, they would inject their DNA and then it would end up killing the bacteria, and then the viruses would replicate that way. So you put this virus and bacteria mixture inside a blender to separate them, and then you put it in a centrifuge to spin it, and the centrifuge would separate the liquid from the pellet. We call it the pellet because it contains all the bacteria. And then you examine this, you look at the two mixtures, and now you see a lot of radioactivity in the liquid. So that's the remnants of the virus shell, the protein coat, which contain a lot of sulfur. But inside the bacteria, there's no sulfur at all. But it does contain genetic information, so it's in there. So now you have to examine batch 2 to put this whole story all together. So batch 2, the viruses were grown in radioactive phosphorus. And the idea is DNA contains a lot of phosphorus. So you let these viruses grow in this solution with a lot of phosphorus, which I'm going to circle there. It's that blue solution. And now you let them do their business. You let the viruses go to work and infect bacteria, and then you repeat the whole process again. You separate them after they've injected their DNA into the bacteria. You put them in a kitchen blender, and after the kitchen blender separates everything, you then put it in a centrifuge and you let it spin, and it's going to separate out the bacteria. And now we actually see radioactive phosphorus inside the pellet. So this would signal to scientists that DNA is what contains genetic information. It's the thing being transferred. It's not the proteins at all, it's actually DNA. And this was apparent because you actually see it in the bacteria because it's been tagged with radioactive phosphorus. Now that you understand some of the history and the origins behind DNA, we can now discuss the structure. So DNA is a helical molecule which contains repeating units of something called nucleotides. Each nucleotide has 3 parts. You have a phosphate group, a ring-shaped sugar known as deoxyribose, and a nitrogen base. There are 4 nitrogen bases. Adenine bonds to thymine, and cytosine bonds to guanine. A purine will always bind its matching pyrimidine, and there's a way that you can remember this to make it easy. So purine, pure as gold. PAG. So purines have A and G, adenine and guanine. On the other hand, you have pyrimidines. So a way that you can remember this is in the pyramids, you see tombs. Uracil, cytosine, thymine. We'll talk about uracil later. That's a nitrogenous base that's found in RNA. So above me are the 2 scientists, Watson and Crick, who in 1953 discovered the structure of DNA was a double helix. So DNA has a helical structure, and the backbone, okay, it looks like a ribbon in that picture right there on the screen. And those blue ribbons are the backbones. Okay, so I'm drawing the trace in there just to show you. So that's the backbone, and it contains sugar and phosphate. So it's the deoxyribose sugar that we spoke about earlier, and the phosphate. And it's just an alternating, repeating pattern of sugar, phosphate, sugar, phosphate, sugar, phosphate. And it happens on the other side as well. So you have sugar there, phosphate, sugar, phosphate. I think you get the idea. So this is what it looks like when it's zoomed in. And you can see the sugar and phosphate. But as you zoom out, you see just the ribbons. Okay, and then obviously they'll coil into chromosomes. Now, what's combining these 2 ribbons, these 2 backbones together, are the purine and the pyrimidine nitrogenous bases, which will pair up, and it's going to bring the 2 sides together. Okay, so you can see there you have guanine binding to cytosine. So you have a purine combining with its pyrimidine partner. And these bonds are being held weakly by hydrogen bonds, which we'll discuss in just a moment. But you can see there, A goes with T. So adenine always binds with thymine. If thymine is on the other side, it's going to bind with adenine on the opposite side. So you can see there. Okay, so it's not restricted to like one side on the phosphate sugar backbone. But do understand, C will always go with G, G goes with C. And that's how it always is. Okay, so that's what you're looking at when you look at DNA. So DNA has this like coiled double helix structure. It's got a sugar phosphate backbone, and it's got the nitrogenous bases in the center, which will pair up, and it's going to bring the 2 sides together. and it coils. And it's known as a double helix. So in short summary, DNA contains genetic information. Several discoveries were made with the experiments involving the mouse and the pneumonia, as well as the kitchen blender experiment, in which you separated the bacteriophages from the radioactive sulfur and the radioactive phosphorus. Phosphorus was found inside the bacteria, and they concluded that DNA is the thing that passed on genetic information, as opposed to proteins. And finally, Watson and Crick put it all together. They determined that DNA has a helical structure made of a sugar and phosphate backbone and nitrogenous bases that pair up and bring the two sides together. Thanks for watching, and I'll see you next time on Wind Biology.